Our next presenter, this is now on Pillar 3, Dr. Nancy Jones, Vice President of Clinical Development from Neuren Pharmaceuticals, to provide an update on NNZ-2591, which aims to improve the impaired connections and signaling between brain cells. Do we have our speaker? Oh, there you go.
I am online.
I'm just realizing that. All right, take it away.
Great, thank you so much. Can you show the slides, please? Oh, great, thank you. Can you go to the first slide, please? I think that's a little halfway through. Well, this is getting queued up. I'm very grateful to be able to be with you today. I only regret that I'm not able to be there in person, and I'm very appreciative of the fact that the organizers have been able to allow me to present remotely. Thank you so much for putting it at the front. So I know that many of you are familiar with our program and our drug, but I know that every year there are new families that join us. So I am going to start by giving a sort of brief overview of who we are as Neuren, what the drug is, and then give you an update on our phase 2 program. So next slide, please. And you can go to the next slide, please. So Neuren is a specialty pharmaceutical company. We are particularly focused on rare genetically based disorders that have a great unmet medical need. And in particular, we are focused on neurodevelopmental disorders. So we have two drugs in development, two compounds, trofinetide and NNZ-2591. And I know the exciting news from this year that many of you may already be aware of is that for our other compound, trofinetide, this year we got an approval for trofinetide for the treatment of Rett syndrome. And that was in the United States by the FDA. And that's with our partner, Acadia Pharmaceuticals. So trofinetide is now available as a treatment called DAYBUE in the United States, and that's available for families for the treatment of Rett syndrome. And we share that, obviously, that's exciting news for us as a company, but and the Rett community. But we also hope that, as you guys know, that when we're doing development of drugs, there are ups and there are downs, and this is one of the ups. So we hope this is also a point of encouragement that things can happen in NDDs for development of treatments. So we are focusing on NNZ-2591. We have developed, we have phase 2 programs in 4 NDDs, and that includes Angelman syndrome. So next slide, please. And just a reminder of where NNZ-2591 fits with other types of drugs that are being developed for Angelman syndrome. And if you can hit next slide, they'll show so NNZ-2591 is a neurobiological therapy, and that means it's something that's focused on improving the function of the brain and the brain cells and the connections between them. So next slide, please. So more specifically, NNZ-2591 is a synthetic analog of metabolite of insulin-like growth factor 1, or IGF-1, and I'll unpack that a little bit. So insulin-like growth factor 1 is a growth factor that's available, that's throughout the body, but it is also in the brain and is responsible for the growth and the maintenance of the brain cells, the neurons, the microglia. And when it functions normally in the body, it breaks up into smaller pieces, which are called metabolites. And one of those metabolites is called cyclic glycine proline. And NNZ-2591 is a synthetic analog of that, which means that it has exactly the same molecular structure as a naturally occurring molecule, with one small change, which makes it more amenable to be used as a treatment. And those properties you can see on the right-hand side of that column. Those are the different properties that are available for NNZ-2591 as a treatment that's going to be used as a medication. So what it is that it is actually able to replicate the same functions as a naturally occurring molecule. So that means it's regulating the amount of IGF-1 that's available. It reduces neuroinflammation. It normalizes the microglia function, and microglia are supporting cells to the neurons, and that helps with the synaptic maintenance, which is like the connections between the cells. And all of this is state dependent, which means that it affects abnormal cells, but there aren't any negative effects on normal cells. Next slide, please. So we have completed our preclinical and phase 1 studies, and this is sort of a brief overview of the findings from that. So in short, we've shown from the preclinical studies that an NZ2591 can have an effect on brain cells, improving structure and function of neurons, as well as levels of IGF-1. And in the mouse models, it's actually showing that there are improvements of those analogous impairments, and that these improvements are seen across a range of symptoms. And that in the phase 1 study, we had a good safety profile and it was well tolerated. Next slide, please. So now I'll talk a little more detail about our phase 2 study. Next slide. So this study is an open label study, which means everyone is getting the treatment. The treatment period is actually 13 weeks, but as you can see from the graph there is a baseline observation period of 4 weeks, and then a follow-up period after the treatment. There is an oral dosing and a liquid formulation, which is taken twice a day, and it includes both in-clinic visits and in-home nursing visits. Next slide, please. So the primary aim is to look at the safety as well as pharmacokinetics. And the pharmacokinetics is how the drug is absorbed and distributed throughout the body. But we're also looking at efficacy, and we're looking at a range of outcomes in this phase 2 trial. So that includes communication, fine and gross motor, behavioral issues, sleep and seizures, GI, as well as daily function. And as you can see, it's a range of symptom areas that we're looking at. And this is our first study in Angelman syndrome. And from the animal model studies, we saw that there was improvement in sort of analogous behaviors across a range of symptoms. We want to look carefully in this phase 2 study, make sure that we're looking and saying, well, what types of things are actually going to improve in individuals with Angelman syndrome? And all of this is going to inform the design of the pivotal trials, the largest trials that would follow. Next slide, please. This is a snapshot of the key inclusion and exclusion criteria. You see there's a range of ages, 3 to 17, as well as a range of genetic etiologies that are included in the study, as well as some specific body weight and the seizure profile requirements. All the participants also have to do assessments and review their current medications and medical history to determine eligibility. So if you'd like to see the full list of inclusion and exclusion criteria, that's listed on clinicaltrials.gov. Next slide, please. The study has been conducted in Australia, and there are 3 centers across 3 cities in Australia. Next slide. And you can do the next slide. Go ahead to the next slide. That's fine. So where are we at now with the phase 2 program? So the study is ongoing. So all the participants are still enrolling and completing their treatment. So the results are going to be reported when all the enrolled participants have completed the study, which is expected next year. And in terms of what comes next, so again, after phase 2, you would have phase 3 pivotal programs. So in terms of that, the plans for a study design and inclusion criteria for a pivotal program is going to be determined once we have the phase 2 completed and we have the full analysis results. Because as I mentioned, we're learning from the phase 2 and to inform what we would do next in the pivotal trials. Maybe follow-up studies will most likely include the US, but it may include other locations as well. And like the study design, obviously, this would be something that will be finalized once we have the phase 2 completed and have the full set of study results. Next slide, please. So if you're interested in learning more about the study, in addition to clinicaltrials.gov, we also have a study-specific website, which you can see here. And then when we do reach study milestones, like for example, getting the results will be a study milestone, and we do make an announcement on our webpage. And so if you'd like to get alerts from that, you can actually go to our webpage, which is neuropharma.com, and you go under news and reports, and then you can click on email alerts and you can sign up for those. And then you'll get, as soon as we make an announcement, you'll get an alert in your inbox if you'd like to sign up for that. Next slide, please. So I'd like to end off to really share how grateful we are to the community for the support of the study, and that includes all of you who are there at the conference and maybe listening online. I wanted to give a particular shout out to Megan and her team at the FAST in Australia, who've been very supportive of our team. Globally, but also our team in Australia who's been running the study, as well as the Angelman Syndrome Association in Australia. Also very grateful, been working from the beginning with the Global FAST as well as the Angelman Syndrome Foundation. And we've been privileged to participate in the ABOM group, which has been very helpful for developing and designing our studies. So at this point, I guess we'll continue with questions during the panel, and I'm looking forward to that. Thank you so much.